So as the final person, I'm going to be repeating some of the themes that maybe you guys have heard from both my colleagues Merrick and Johanna. I think you'll see how probably our ideas are bouncing off of each other as we sit next to each other in the lab on a daily basis. But I'm going to be applying this work to individuals with eating disorders. So I specifically look at individuals with anorexia nervosa and atypical anorexia nervosa. So much of this work, actually, I'm sure like many of us sitting in the audience, actually stems from my work as a clinical psychologist. So as you may have heard, that I'm a trained clinical psychologist and neuroscientist. And a lot of what I try to think about is how could I actually improve treatment and therapy for patients with eating disorders? And that's because many of the patients that I see with eating disorders, I often see, unfortunately, are relapsing, meaning that they come back into my office over and over again. The time that they spend in treatment is oftentimes actually upwards of 20 plus years. So it means the course actually of treatment in eating disorders is extremely attenuated. The other component to this is that individuals with eating disorders, we don't actually have any FDA-approved medications, and that's across the entire world. So it means the only option really that we have right now for individuals with eating disorders are therapeutic approaches, which is quite challenging when many of these approaches are actually working against the biology, which you'll hear about. So one of the things that we've been trying to think about is what are state-specific related changes that we could target where we could identify a moderator, meaning that are there changes that we see that are closely tied together that happen as a result of the eating disorder that we could identify different targets to actually improve treatment, reduce the time someone is spending in treatment, or permit relapsing effects. So today I'm going to be walking through these for you. I'm going to talk briefly about this gray matter volume and body mass index relationship that we see over and over again in anorexia nervosa, and hopefully explain to you how closely tied together this is. Next, I'm going to be talking about a similar approach, which you just heard from my colleague Johanna, about targeted proteomics and gray matter volume and some of our initial results that we've now replicated a few times. And then I'm going to go through some preliminary results from our research study. This is unpublished work as well. I'm going to talk about how to apply this to clinical practice, and then talk about where we're hoping to go next. Just as a reminder, though, I think it's helpful to paint the landscape, particularly of eating disorders. It's oftentimes an understudied psychiatric disorder that's oftentimes referred to as hidden or forgotten about. So worldwide, about 41.9 individuals were diagnosed with an eating disorder. This is work done in 2019. We know that after the COVID pandemic, actually this is one of the disorders that has skyrocketed, and there's numerous hypotheses for why this might be. To put this in perspective, though, this is just shy of the entire population of Poland or Canada, or over actually the population of both Poland and Canada, and shy of the population of Argentina. So the global prevalence is around 7%. Eating disorders remain oftentimes one of the highest and most lethal of all psychiatric disorders. So the prevalence rates are actually similar to that of bipolar disorder, ADHD, and neurodevelopmental disorders. In the United States, where I practice, the prevalence is around 15% in adolescent females. So in the United States, it does seem like the prevalence could be higher, although we don't think this is necessarily due to reflecting the prevalence in other countries. We think that it's oftentimes related to an overlooked and understudied disorder. My work focuses primarily on restrictive eating disorders, namely anorexia nervosa and atypical anorexia nervosa. Anorexia and atypical anorexia nervosa, they're virtually the same when we think about morbidity, mortality, as well as the factors that might contribute to the risk and development of them. The only difference between them two is, for whatever reason, and we don't know, there's a lot of hypotheses about this, is that individuals with atypical anorexia nervosa, for whatever reason, their body mass index remains this demarcated value above 18.5, which means that they are operating at a body weight that is supposedly healthy, so to speak, but they're engaging in the same type of behaviors. That would be extreme calorie restriction, this intense deep fear of weight gain, and a skewed perception of body image, weight, or shape. So we know anorexia, it affects the entire body, right? So one of the most challenging actually components about treating this disorder, as individuals begin to restrict, it begins to actually break down their entire organ system. One of the most striking factors that we've seen over and over again is actually revealed in MRI images of individuals with anorexia nervosa. And it's so striking, you can actually see this sort of to the naked eye, so to speak. And this is this decrease in gray matter volume that you really can observe in that cortical thickness. So you can actually see that arrow pointing there that's demonstrated by that larger cortical thickness that you would see in the image on the left. And this has been shown over and over again, but no one exactly, we think it's attributed probably to this weight loss that occurs, because we do see this actually restore over time. So this is a relationship that's been showed pretty consistently now, that we see these reductions in gray matter volume, cortical thickness and surface area in individuals with both severe anorexia nervosa, and then once these individuals begin to recover, later on down the road, we do see restoration of this. What's hard is that to get someone into treatment, right, we might be able to say we should work on gaining weight, but that weight gain period in anorexia nervosa might take the entire therapeutic time period that you have with someone. We're talking 40 weeks of treatment where someone's coming on week to week. What's challenging then is that these reductions in gray matter volume are actually linked then to the cognitive decline that we see in anorexia nervosa, and which is both necessary for someone to engage in treatment. So you'll hear over and over again the story that someone is really poorly engaged in treatment for eating disorders, right? They're not thinking clearly, they even discuss sort of the brain fog that they might actually describe, or they're uninterested in treatment. Well, it could be, and what we think it could be, is because of this sort of extreme weight loss actually does result in these gray matter changes that we observe over and over again. And restoration of that does happen, but it happens basically after they're out of therapy, right? So that would mean that someone would go to therapy, gain the weight, and then perhaps 6 months later, a year later, when the restoration could occur, only if they're staying at that weight. So one of the things that we've been trying to identify is if there's certain things that are really tied to this gray matter BMI relationship that could act as moderators and thus potential drug targets. So we could actually use these changes that are happening in the periphery as actually new drug targets themselves. So we've seen then exactly this positive relationship between BMI and cortical thickness. The other thing that we don't know is that in some individuals, where actually the recovery is occurring does differ. So what you'll see oftentimes in people who are partially weight restored or just at the end of weight restoration is that this reduced gray matter volume actually persists in both the thalamus, the putamen, the amygdala, and the hippocampus, which I'll come back to at the end and why this is so important for patients. So recently, we've been in the search for moderators, and what we've done is we tried to think about, are there state-specific changes that are really, really tied to BMI among individuals with anorexia nervosa? My colleague at Karolinska Institute over here on the left, Ida Nilsson, she had shown in 2021 that actually anorexia nervosa is associated with a widespread inflammation sort of profile. So instead of just looking at single protein levels, as Johanna mentioned is sometimes challenging to do, is what she did is she used that same sort of targeted proteomic approach, which actually sequences 92 different inflammation markers to see if there are inflammation markers that are closely tied to BMI. And what she showed is yes, in fact, individuals with anorexia nervosa are associated with widespread inflammation throughout the whole body, which probably makes sense, but actually it was oftentimes opposite of what one would expect for anyone that researches obesity, right? Because obesity is also associated with widespread inflammation. But what we know genetically, right, is that both inflammation, or that actually anorexia nervosa and obesity genetically represent opposite ends of the bookend when we think about metabolic impacts, meaning that the same thing that's maintaining someone's weight with obesity is the same gene that's actually operating on anorexia nervosa, meaning that people are sort of stuck at these extreme ends of their weight. So in some ways, the protein expression level, it would make sense that you'd see this aberrant inflammation in both anorexia and obesity. This was done in adults who had both active anorexia and who had recovered from anorexia nervosa. So more recently, my colleagues and I had actually replicated this in a sample of individuals both with anorexia, this atypical variant, which was important because it allows us to look across the entire weight spectrum, who are adolescents and young adults. So we do still see this in this younger adult population. So we focus then on cytokines, right? Because these are sort of these sort of second-level moderators that we actually might be able to target in the periphery. And they play a significant role both in mediating and regulating immunity and inflammation, right? So when we think about cytokines, it's not necessarily that all of them are bad or all of them are good. It's in fact the interaction of them together that actually creates this sort of adaptive immunity and positive immune system for regulation. So in this preliminary data that I'm going to show you now, we actually aim to show that these cytokines, which I hope I've shown you are important to both brain health and are linked to inflammation, may impact gray matter in a state-dependent manner. And we hypothesize that peripheral inflammation would moderate this BMI-gray matter relationship, because that would allow us to sort of begin to build this evidence that there could be sort of markers in the periphery that could be targeted to impact this gray matter relationship that we continue to see. So to do this, we had individuals with anorexia nervosa and atypical anorexia nervosa who were all about the ages of 18, so a little bit younger, a little bit older than that. We had 88 individuals, so you'll see that the sample sizes in these studies are significantly smaller than my colleagues in schizophrenia, but we are trying. Then we collect T1 imaging scans of them. So rather than looking at white matter, though, we're looking at gray matter, and this is because this is this most consistently replicated finding in anorexia when we're thinking about what is tied to body mass index. And then we process them with the standard pipeline in FreeSurfer to extract the 3 gray matter indices shown there in the bottom left-hand corner. So cortical thickness, surface area, and then the composite one of both cortical thickness and surface area, which we consider gray matter volume. We then collect a blood draw around 9 a.m., so that allows us to actually control for the circadian rhythm, which also has a large impact on the proteomic measures themselves. So that way we can look at just the proteins when everyone is in a fasted state, which also has an impact on proteins, and then only in the morning, so we're controlling for that circadian rhythm. And then we sequence them using this target inflammation panel. So just, I think, as a reminder, why in the world are we continuing to focus on proteins themselves? I think the big thing is that proteins are actually sort of, when we think of the central dogma, are at the top, right? So when we think about genetics, we think about RNA, protein screening, and then the phenotype, right? Proteins are actually closest to that phenotype, which means that they reflect the change over time. So when we're interested in state-dependent moderators, we need something closest to the change. That's why we aren't focusing on RNA or DNA sequencing in this, right? Because we're interested in a state-dependent marker that we could modulate with potentially another drug, which is why we've continued to focus on proteins. They're dynamic. So as I mentioned, we do a lot of things in these studies to try to control for actually the protein expression level, because they're changing over time during the day related to what someone's actually eating oftentimes, if they're in a fasted or a fed state, which means they're challenging to collect because they actually are reflective of sort of an entire whole body phenotype or expression or body state. The other thing is that they actually provide the most actionable targets for therapeutic interventions, which for me, studying eating disorders is extremely important since we actually don't have any FDA-approved drugs. So as I mentioned, we use this O-link target inflammation panel. We do a whole bunch of different QCing, which basically just means we try to actually make sure there's not any one protein that's spiking too high, and then when we harmonize it with other ones, that they're all looking similar. Next, what we did is we actually took both sort of a exploratory approach, but actually let the biology first speak to us for how we're grouping these together. So we grouped the proteins together based on the cytokine families, right? So rather than looking at IL-10 specifically, or leukemia inhibitory factor specifically, we're actually grouping these together because each of these cytokines is related to a larger family that has a similar receptor downstream. What we do after grouping them biologically is then we check statistically, right, using a confirmatory factor approach to say that, like, basically do our expression levels actually hold together to make sure that they're truly sort of families. So we take both an approach to say, let's actually use the biology and apply that to these proteins so we make sure these families are together, and then statistically do our results hold. And what's great is this approach seems to work pretty well. So the statistics and the biology actually are a complete interaction here. So these are our different protein families that we're focusing on. So we have the IL-10 family, the IL-6, TNF family, and then 2 chemokine families, the CXC and the CC chemokine family, and then TGF family. And then the individual proteins you can see are listed there. Next is what we wanted to do is we wanted to look across to make sure we have a wide enough distribution of BMI to actually look for moderating effects of this. So on this graph here, you can see on the x-axis are our groups. As I mentioned, there's that AN group in the middle. That turquoise is the atypical AN group. And then in yellow is the healthy control. And what you can look and see from that is that the AN group definitely has that wide, really low BMI range. But that atypical group and that healthy control group do overlap enough, which means we have sort of spread the entire BMI spectrum, which is important for actually identifying these moderators itself, which means that we don't have sort of like two separate groups, that actually we can look continuously, which allows us to look for moderators, or if we want to look at mediators, we could. We then use a set of linear regressions to look at a moderation analysis, which allows us to look at how cytokine family impacts the BMI gray matter relationship while controlling for skull size. And then we use a moderation analysis using linear mixed effect models, so that way we can actually control for hemispheric differences between the two different sides of the brain, skull size, and then we used a stepwise approach to additively include sort of environmental variables that we know impact these proteome variables as well. So we do similar things that Johanna mentioned, where we are controlling for people that are smoking, the age that they were at the time of the analysis, comorbid psychiatric conditions to say, is it truly related to anorexia themselves, as well as any psychiatric medications they were taking at the time. So we use a stepwise approach to say, of these lifestyle factors, which one is contributing the most variance to our model, and then that way we actually are including those in the model themselves, rather than putting them all in the model, which would likely lead to overfitting. So first, these results just show you the relationships between BMI and gray matter volumes. This is simply a replication, basically, to make sure that in our data, are we truly seeing this really strong association between BMI and gray matter that's been replicated. And what you can see here is that, yes, we do see that. It's significant in all three indices, cortical thickness and surface area, as well as gray matter volume. What is interesting, though, is that surface area continues to be sort of the weakest and have the smallest effect size out of all of these, and that's been replicated over and over again. And we think that's oftentimes related to the fact that anorexia develops at a younger age. Here we're actually just testing to make sure that none of our proteins are mediators, not moderators. So this allowed us to actually continue forward with the moderation analysis. This is simply showing that none of them are mediators. You'll see that TNF family actually does come up. So this is the relationship between the TNF family and gray matter volume. We ended up being able to exclude this as a mediator as well. Okay, so I'm going to walk you through after we did our linear mixed effect models and moderation analysis, the main family that I'm going to continue to talk about, because this is actually the only one that comes out across all results, is this CXC chemokine family. And what we see is that over and over again, the CXC chemokine family actually attenuates the relationship between BMI and Z-score and gray matter volume. So in that blue line right there, these are individuals who have low levels of that CXC chemokine expression. And in the red line, these are individuals who have high levels of the CXC chemokine family. So you can see when expression levels of the CXC chemokine are particularly low, so this is below the 16th percentile, then it means that it attenuates the relationship. When they're high, you see this flattening effect. And the high ones, just as a reminder, are at the 84th percentile, so they are truly significantly higher. What we then did is, since we looked in sort of the overall gray matter results, then we looked within specific lobes, and then next within the specific regions. And what we see is that this chemokine family, it attenuates the BMI gray matter relationship in the temporal lobe, the occipital lobe, and the frontal lobe. Within those regions, so within the frontal lobe, we actually see it seems to affect the medial orbital frontal lobe, and in the temporal lobe, it's affecting middle temporal regions. And so again, this is just here with the graphs here. As you can see again, that red line is individuals who are extremely high in the CXC chemokine expression, and the blue line there is individuals who are low. And you can continue to see that the relationship is attenuated. So just to summarize this, we continue to see this really strong BMI and gray matter volume relationship. It seems like the peripheral CXC chemokine levels are an important moderator of this relationship, which might lead to us thinking about different ways to actually target these CXC chemokine levels that could both be more appealing to individuals with anorexia nervosa rather than saying, hey, come into treatment, we're going to work on rapid weight regain. That means everyone's just going to walk right out the door. No one's going to actually be enrolling in treatment. And we know that these chemokines are crucial for regulating inflammation broadly. So none of what I've showed you is actually anything molecular or cellular. But just for a moment, let's think about this, because I think this is an interesting thing. So both CD4 cells and these T cells are actually regulators of metabolic function and neuroinflammation. When I say metabolic function, both of these cells actually, they're considered sort of the memory cell of metabolism, meaning that they are holding these individuals at the spot of what their body weight might be. They release these macrophages and have an impact on astrocytes, which where is that CXCL expression is actually in the middle, right, which is actually influencing the expression of neutrophils, releasing the elastase, and could be breaking down that blood-brain barrier functioning. So again, kind of even going back to what Merrick has been saying, it seems like it could actually be breaking down sort of and changing the way the blood-brain barrier is actually functioning. We're doing some studies right now to try to characterize that. So this could be why we see this increase in CXC chemokine expression in anorexia, which sort of might be suggesting this prolonged immune response and may lead to our observed attenuated relationship in the BMI and gray matter volume association. Another point that's interesting to think about, we're not looking at these inflammation markers independently, but when it comes to drug targets, that actually might be an avenue that we would want to continue to explore. So there are several that we found that there are actually, that are all ligands for the CXCR3 receptor, and they're currently actually working on drugs to target this as well. They could be potential treatment strategies for anorexia. One thing, though, is I think that research on this area we know will be slow and long, right? So we're talking about 20, 30, 40 years before we actually are thinking about a drug that could be working. So I always like to think about what actually could I tell the patients that I work with, or what could you share with your patients right now about this research that could be either motivating to stay in treatment or help them sort of understand what's going on. One, I think that the big one is that actually this BMI gray matter volume relationship, especially for adolescent patients as well as their parents, is really motivating to help them actually stay in recovery. So oftentimes people will come into treatment and they won't want to gain weight, but they'll tell me that they have a sort of a brain fog, that they can't figure out how to lift, right? Or that they can't think clearly anymore because they're constantly thinking about food, or they're constantly thinking about what they're going to eat next. And I help them understand that actually because they're at this extremely low weight, if they stay at that low weight, then the gray matter itself in their brain is never going to be able to restore. And we'll call that sort of brain health, maybe even potentially really broadly, right? To help them recognize that actually what they're doing by staying at this low weight and resistant continuing to gain weight actually might be contributing to these symptoms that they're experiencing that they are motivated to change. Things like brain fog or an inability to think about different things, right? Because we know gray matter volume is actually key for anything related to set shifting or executive functioning. The other thing that oftentimes I'll share with them is helping them understand that, yes, treatment is hard and be empathetic around that, right? So actually what we're asking them to do in treatment is kind of in some ways asking them to overcome their current neurobiology, right? So these regions that we see are extremely affected in eating disorders and could actually be continued to be affected when they're in this low weight state driven by inflammation, are areas of the brain that are related to taste and feeding behavior. So when they say they're not hungry, they're not interested in eating, that could actually in fact be true. And instead of dismissing that, right, it's helpful to even for families and patients to say, yes, I do actually understand this. We do have evidence that shows that. I understand that you're not hungry right now. I understand that you're not interested in food right now. But bear with me as we try to work through this as well. It also kind of gives them some hope too to think about that these things can restore as well over time. And the other areas of the brain, right, are related to appetite dysfunction sort of more broadly. So when people say that they're not hungry or complete food avoidance. So just to summarize these study results just one more time too. So what we see is the CXC chemokine family, it does seem to be the key one that actually reduces this BMI and gray matter volume relationship. It seems to be isolated potentially in the medial orbital frontal cortex and medial temporal lobe regions. Moderating the CXC chemokines may indirectly support gray matter restoration, promoting neuroplasticity and supporting behavioral interventions, and therefore hopefully optimizing treatments for anorexia nervosa. There's a couple key limitations I'll just call out. One is this is a cross-sectional study, which is extremely challenging to draw concrete conclusions about. It is a relatively small sample size. That's why I talked about we do a lot of different things to actually control methodologically before we start collecting data rather than statistically, so we try not to overfit all of our models. We are limited to just these proteins, right? So there's 100,000 to 400,000 human proteins in the body as well. We're looking at 92. So of course there could be more that we could not be thinking about, but we started with more of an inflammation hypothesis-driven study as well. And then lastly, these are all peripheral proteins, right? So they're getting actually inputs from all of the organs that I talked about that are being affected. So they could, right, not actually be most related to the brain. So we are working right now on at least getting a little bit closer to central nervous system by collecting CSF in individuals with eating disorders to actually look at the proteins expressed in the CSF. And last, I just want to thank, these are our clinical research coordinators who are all excitingly going on to the next stages of their career coming up. So we're hiring a lot of new people. And they've played a significant role in both the data collection and analysis of these studies, as well as the entire team. So special gratitude to my mentorship team up here on the left, as well as the entire eating disorder. This is the Clinical Research Collaborative, so it means all these individuals both see patients and actually work with the data, which is vital when we're working with all patient-collected data, as well as the funding resources listed on this slide. Happy to take a few
Well, thank you.